Hi, my name is Ling. I'm a research fellow from National University of Malaysia. The title of my presentation today is Optical DNA Biosensing Methodologies for Clinical Diagnostic Application. Nowadays, the demand for DNA detections has increased, mainly for the understanding of various biological processes and developing biotechnological devices due to their ultra-sensitivity and real-time detection regime compared to traditional hybridization assays that involve tagging the DNA probe with radioactive or fluorescent labels that are time-consuming and hazardous. Generally, the information on DNA hybridization can be acquired by utilizing small molecules such as drugs and ligands that are able to bind to single or double-stranded DNA through electrostatic interactions at various mechanisms, including group binding, alkylating, and intercalating of DNA. I'm going to share several optical DNA biosensors which have been developed in our group with different fabrication designs and sensing methodologies. An optical genome sensor based on shift-based complex DNA label and acrylic microsphere as polymer supporting of the capturing DNA probe specific to dengue virus as a model was developed by reflectance spectrophotometry detections method. The shift-based zinc-sulfine complex was synthesized by reacting 2 equivalent of 2,5-dihydroxybenzaldehyde with 1 equivalent of 0-phenylalanine diamine through a simple 1-quart reactions in the presence of zinc acetate in ethanol solvent under reflux conditions with continuous stirring for 5 hours. The aminated DNA probe was attached to the succinimide acrylic microsphere by a peptide Ling. The synthesized metal-sulfine complex was exploited as a synthetic DNA binder, which could intercalate between DNA bases by pi-pi stacking interaction between the pi electron delocalizations in nucleobases stacked aromatic ring and azomatine as well as benzene group of the zinc labels and functionalized as optical DNA labels. The bare acrylic microsphere gave the highest reflectance intensity due to its bright white sphere background that reflected all 7 colors of the rainbow spectrum, and upon covalent immobilizations of the amine functionalized capture probe through the succinimide modified acrylic microsphere, the DNA biosensor response shows slight decrements in terms of reflectance intensity as it absorbs some photon from the feed fiber optical light. However, the DNA biosensor exhibited no color change and remained white in color. Hybridizations of the immobilized DNA probe with target strand followed by intercalations of the immobilized double-stranded DNA with optically active zinc shift-based complex, the DNA biosensor displays an obvious change of color from white to light yellow and attenuated the light reflectance intensity as darker objects absorb more photons than lighter color objects. Therefore, the proposed novel DNA bar up top permit naked eye observations of dengue virus as it gave distinct yellow colorations on the microsensor surface, and because the acrylic microsphere is hydrophobic in character and that could hinder non-specific interactions with the polarizing sulfine label. Characterizations of reactions with DNA of the optical DNA biosensor with reflectance transducer has clearly shown the role of zinc-sulfine optical properties in showcasing the selectivity of immobilized DNA capture probe towards its target DNA, in which it gives the highest reflectance intensity compared to non-complementary and one-base mixed-match DNA. The highest recognitions of the immobilized capture probe towards the target DNA was attributed to the maximum base pairing between 16 complementary bases of immobilized DNA probes and target DNA, that enabling the full hybridization process to take place in order to use the immobilized double helical structures of the double-stranded DNA. A sigmoidal response trend of the reflectometric DNA biosensor was obtained in determining a series of dengue virus DNA concentration ranging from receptor to centimolar concentration, as expected by increasing the concentrations of dengue target DNA from femto to millimolar. The reflector response increased proportionally as the yellow coloration gradually built up on the microbiosensor surface. This was attributed to the growing number of the immobilized DNA duplexes formed on the DNA biosensor surface, whereby promoting a higher number of yellow color shift-based complexes intercalated into the immobilized double-stranded DNA, and that reduced the amount of the light reflected from the darker DNA biosensor background. The response curve of the DNA biosensor was eventually leveled off, from centimolar cDNA and onwards due to the saturation phase that's achieved on the DNA biosensor surface. The optical DNA biosensor provided a new approach in the diagnosis of dengue fever, whereby the detections of dengue virus serotype 2 genome extracted from clinical serum, urine, and saliva of dengue virus infected patients under informed consent have been validated with RT-PCR methods. The PCR analysis has been done for blood, urine, and saliva samples collected from patient 1 to patient 4 to confirm the presence of dengue virus serotype 2 in this sample. The PCR electrophoretic result showed that the human fluid sample from patient 1 to patient 4 were all positive, reasoning with approximately 69 base pair of the expected amplicon band, demonstrating the presence of dengue virus serotype 2 in this sample. And in order to determine the accuracy and reliability of the developed optical DNA biosensor for dengue virus detections, the result obtained from the DNA Optical were compared with and established standard method or RT-PCR for nucleic acid detections. As summarized in Table 1, the t-value obtained for all sample types are less than the critical t-value of 2.776. Hence, there were no significant difference in the dengue virus serotype 2 cDNA concentrations detected by both measurement methods, whereby the analysis of actual DNA obtained from dengue virus infected patients was shown to correspond perfectly with the standard RT-PCR method with respect to DNA quantifications of dengue virus in human biological fluid sample. Consequently, the proposed refractory DNA biosensor would offer a non-invasive approach for the dengue diagnosis. Because pipeline compounds such as these coupled to phthalocyanin are ligands for human telomeric G-quadruplex. Here we develop an optical RNA responsive G-quadruplex biosensor with a long loop which is complementary to the target DNA. As a proof of concept, the functional G-quadruplex DNA is designed with a 30-mer loop targeting the dengue virus serotype 2 RNA sequence and immobilized on the ecliptic microsphere. Hybridizations between the loop on the G-quadruplex structures and the dengue virus serotype 2 RNA will cause the G-quadruplex to unfold and release the bound ligand compound, which refers to the nRNA coupled to phthalocyanin, a highly specific G-quadruplex ligand, and leads to a color change of the RNA biosensor surface that allows visual estimations of the presence of dengue virus serotype 2 RNA for direct colorimetric diagnosis of the dengue virus infection. The principle behind the RNA sensing using G-quadruplex is that in the absence of the target RNA, the G-quadruplex maintains the G-quadruplex structures and thus the G-quadruplex ligands remain bound due to its square planar geometry and aromatic ring structural properties, and G-quadruplex biosensor shows blue colorations to our naked eyes. In the presence of the target RNA, the G-quadruplex structures unfolds, releasing the G-quadruplex ligand and rendering a distinct color change of the G-quadruplex biosensor. This G-quadruplex is designed to target the specific sequence of dengue virus serotype 2 RNA. The G-quadruplex sequence is based on the human telomeric DNA sequence. The G-quadruplex probe contains a 30-mer length loop in place of the second TTA in the HTML DNA that is complementary to the target RNA sequence. Importantly, the target sequence contains an AACCC at the fifth end, as it is expected that these 5 bases would hybridize with the third GGG and the adjacent TT of the G-quadruplex, leading to efficient unfolding of the capture group, since the GGG regions is involved in the formations of the G-quadruplex. These are some preliminary characterization studies of the optical RNA responsive G-quadruplex biosensor. A low-level reflection response of the G-quadruplex biosensor without the presence of the RNA was attributed to its blue colorations background, as the immobilized G-quadruplex maintains a blue background, which is the G-quadruplex structures with its G-quadruplex ligand remains bound. The G-quadruplex biosensor show an enhanced optical response after hybridized with target RNA due to the unfolding of the G-quadruplex followed by releasing the G-quadruplex ligand and resulted in color fading of the G-quadruplex biosensor. The optical G-quadruplex biosensor response kept increasing, as increasing the RNA concentrations from zepto to nanomolar concentration level with the blue colorations of the G-quadruplex biosensor to slowly fade out as a result of the increments in the optical ligand compound releasing rates from the G-quadruplex biosensor. This is another DNA microoptope developed based on sandwich DNA hybridization strategy. The DNA microoptope is designed here as an optical sensor based on DNA recognition probes and sub-micrometer size gold particle label polyelectrolyte coated latex particle as the optical label, and succinylate functionalized acrylic microsphere as the DNA hybridization platform. The latex microsphere was modified layer by layer with polyelectrolyte to form multi-layer latex particle before being electrostatically attached with the gold nanoparticles and chemically grafted to the signal probe by thiochemistry to produce a gold latex reporter label specific to the target nucleic acid sequence. The acrylic microsphere was conjugated to the amylated DNA capture probe via the peptide covalent bond. The DNA sandwich detections procedures were carried out by pre-hybridizing the gold latex optical amplified reporter label with target DNA prior to hybridizations with the immobilized DNA probe, thereby bringing violet colorations to the acrylic microsphere DNA supporting matrix. The FESAM image shows the spherical morphologies of the acrylic microsphere. The spherical morphologies of the polyacrylate is essential in the immobilization process as it maximizes the surface area available for the DNA probe immobilization while covalent binding. The FESAM micrograph of latex colloidal particle presents a rather homogeneous size distribution. The small size of the latex nanosphere provided a large specific surface area for high loading capacities of the gold nanoparticles optical amplifier reporter label through electrostatic interactions in order to generate a substantial optical amplification effect on the reflectance response upon sandwich DNA detections of the target DNA sequences. Reversibility study was conducted to determine the reproducibility of the optical DNA biosensor for assay of the DNA after treatment with a mild sodium hydroxide regeneration solution. The DNA biosensor was capable of giving a repeatable reflectance response for 3 consecutive DNA detections, whereby a substantial decline in the relative reflectance signal of the DNA biosensor was observed starting the 4th cycle of the DNA analysis, which was attributed to the dissociations of the hydrogen bond between nucleobases of the DNA double helix, as well as permanent disruptions to the DNA probe strand from the acrylic immobilization micromatrix after repetitive regenerations of the DNA biosensor with the sodium hydroxide solution. As such, the sandwich type DNA microoptope based on the gold latex sphere label can be reused for optical detection purposes up to a maximum of 3 times. Current clinical diagnosis of dengue virus infections in humans is based on serology tests that involves a virus isolation in cell culture. Therefore, it must be performed in the laboratory with necessary infrastructures and technical expertise. Furthermore, detections and classifications of dengue virus serology tests usually takes several days or maybe longer than a week or 2. Non-structural protein antigen detection kits are commercially available and can yield results within a few hours. However, the assays are not type-specific and require specific equipment and expensive. The rapid kits based on detections of IgM antibody in human serum will normally require 5 days for the patients to produce a detectable level of the anti-dengue antibody before the rapid kit can be used. Nucleic acid detections derived from nested or semi-nested PCR, RT-PCR, and RT-qPCR for amplifications of target sequence are, however, possess drawbacks such as cross-contaminations, as they use several types of primers, requires high-quality assurance lab and assortments of DNA extractions and purification kits for a more reliable and a detailed quality control in order to improve the overall specificity. In general, the currently available commercial technology employed in the dengue virus detections appear to be laborious, low sensitivity, expensive, and invasive, as blood sample is usually required. The proposed optical DNA or RNA biosensor can be employed as a solution to this limitation based upon semi-quantitative visual color inspections of dengue virus DNA or RNA immobilized on the DNA biosensor, as the DNA probe is selective towards its complementary target DNA or RNA. Thus, visual monitoring of dengue virus in non-invasive human bodily fluid, for example, urine and saliva sample, will permit rapid diagnosis of dengue infections in humans as fast as the first day of illness onset. As a conclusion, we are the first to present this new optical DNA biosensor model based on the binding and interactions of the transition matter complex and gold latex sphere as the optical DNA label. and the solid-state DNA hybridization chemistry on the alkaline microsphere were investigated for the first time with refractometric methods. The optical DNA biosensor system shows single-base mixed selectivity and sensitivity down to zectomolar level. The color transitions matter complex and its DNA binding ability allows naked eye detections of target DNA, which has high potential to find practical applications in various clinical diagnostics, including diagnosis of genetic diseases, drug screening, forensics analysis, and detections of emerging infectious diseases, as well as for identifying bacteria or pathogenic or microorganisms in human foods and environmental samples. With this, I end my presentation here. Thank you.